Rh5 as a relative. And we thought all of this together, the fact that it seems to be an essential interaction for invasion, it seems to be used across strains, and it's a relatively conserved protein, made it a good vaccine target. And multiple other labs have worked on Rh5 as well, and it's now the focus of vaccine studies and entered phase 2 trials, that work is ongoing. But also, you don't have to understand a protein or understand an interaction to think about it as a vaccine target. So we've also been involved in systematic screening, just expressing panels of recombinant proteins in the best, in the most folded and functional manner that we can, raising antibodies against all of them, and then screening for whether they block invasion. And it's critical, I think, when people do this, that they take strain diversity into account, because all Plasmodium vaccines will struggle with genetic diversity. There's a huge pool of genetic diversity out there in the world. And so we'd like to incorporate diversity at the earliest possible stage in our screening. So this is from a paper published a couple of years ago, raised antibodies against a panel of different proteins. You can see Rh5 clearly inhibits invasion in both strains, but we identify other proteins that can do so as well. And we test those proteins both alone and also in combination. So this is a sort of systematic reverse vaccinology, it's called, going from the protein to candidates approach to identifying new malaria vaccines. And we're now doing this exactly the same thing for Plasmodium vivax, which of course causes the majority of malaria outside Africa. But with vivax, you have the additional complication that you can't grow the parasites in vitro. So you have to use some model systems. And in this paper we published earlier this year, we used Plasmodium knowlesi as a model system, both just wild-type Plasmodium knowlesi, which worked much better than we thought it might, but also Plasmodium knowlesi, which had been genetically modified, where we'd replaced Plasmodium knowlesi genes with Plasmodium vivax genes. So we, again, raised antibodies against a panel of Plasmodium vivax proteins, and then tested them for their ability to block invasion in either knowlesi or modified knowlesi. And we're now expanding that work out to more than 100 proteins at the moment. But most of my talk today is actually not about individual proteins at all, because essentially if you're trying to identify new vaccine targets or new intervention approaches, you take clues anywhere you can get them. So you take clues from an interaction you've identified, you take clues just from systematic screening without thinking about what the biology is, but you also take clues from other sources. And we, last year, completed a massive amount of work with colleagues around the world that followed up one of those clues. And that clue came from human genetics. So human variants have long been known to protect against severe malaria or infection. And there are very well-known examples. Sickle cell hemoglobin, for example, which is present in high frequency across West and Central Africa and significantly reduces the amount of severe malaria. Duffy negativity, which largely or almost completely protects against infection with Plasmodium vivax. And large-scale genome-wide association studies are now identifying new variants associated with protection. And it's an interesting story that parallels what's happened with genome-wide association studies for other diseases unrelated to malaria, where early on you see a few very strong hits, but then as you go deeper and deeper with more and more sample, you see other hits, which might still be strong, but they're just much rarer. And this is an example. So Dominik Kwiatkowski's lab at the Sanger, who's really led this GWAS work for malaria, some years ago, they identified a strong hit associated with protection against severe malaria. And this is the Manhattan plot that shows the location of the SNP, which is most strongly associated with protection. And it sits right next to a cluster of genes that are known to be involved in invasion, the glycophorins. Actually, for a long time, we referred to this SNP as the FREM3 SNP, because it's actually closer to another gene called FREM3 than it is to these glycophorins. And FREM3, as far as we knew, had nothing to do with malaria parasite biology. So it was a bit of a mystery. And then amazing work from Alan Leffler and Gavin Band in Dominik Kwiatkowski's lab, using a combination of short read and long read sequencing, revealed that in fact, all the SNP was tagging a much more complicated large-scale DNA rearrangement in the glycophorin cluster. And that rearrangement actually takes what are 3 related genes and completely splits them all around so that you have, instead of 3 genes, you have 5 genes, You have a wild-type copy of glycophorin A. You have two copies of a hybrid gene that has bits of glycophorin A and bits of glycophorin B. And then you have two copies of glycophorin A, but where the regulatory regions are all scrambled and messed around. And this is just one example. It's actually this example here, which is called DUP4, duplication 4, of multiple large-scale rearrangements, duplications, deletions in this gene locus. They're essentially found in Africa and nowhere else in the world. And different ones of the different of these rearrangements are more common in different parts of Africa. And so far, only this one, DUP4, has been associated with protection against malaria. But it seems likely, and I'd be willing to wager a small beer or so, that some of these others will be associated with protection with malaria over time, just as we get more samples in these GWAS data sets. So this variant appears to be restricted to East Africa. It's found at its highest frequency on the coast of Kenya, where it reaches a minor allele frequency of 10%. So that means 10% of people have one copy of the rearrangement and 1% of people have two copies of the rearrangement. But if you have two copies, actually, the level of protection is quite high. So you're 70%, there's a 70% reduction in your chance of getting severe malaria. And that's about the same level of protection provided by sickle. And interestingly, this allele, this rearrangement, is more common in this population than the sickle allele is. So although over the whole of Africa, it's vanishingly rare, in this region in East Africa, it's actually as common as sickle is, if not more common, and provides as much protection as sickle does, and also much more protection than the current best malaria vaccine. So we became involved in a collaboration with multiple other people that I'll introduce in a minute, trying to understand how this rearrangement protects people against malaria. And it really was an epic study. So we screened more than 600 children along the coast of Kenya, looking for individuals who had either a single or double copy of this rearrangement, collected red blood cells from those individuals, and then put them through a series of different assays, which I'll talk briefly about in a minute. And it's really important to emphasize that this was a massive collaboration. So the genome sequencing work had been done previously by Dominic and Gavin and Alan, and the work on the functional follow-up was led by three people: Alex Marin Menendez in my lab, Sylvia Kariuki, who was in Tom Williams' lab, and a PhD student called Viola Introini, who was in Pietro Cicuta's lab in Cambridge. And a large number of Kenyan researchers and collaborators were involved as well, and also Mike Weeks, who's as a PI in my new institution, the Cambridge Institute for Medical Research. So the first thing we did was just look at the red blood cells themselves and asked if there was anything different between the DAN2 wild type and the DAN2 heterozygote and DAN2 homozygote cells. And through a combination of flow cytometry and surface proteomics, using this wonderful technique that Mike Weeks had developed, where essentially you label and purify surface proteins and no other proteins, we could see there were a number of different changes on the surface of these red blood cells. So a few proteins were down, a few proteins were up. This is a comparative plot comparing the abundance of proteins on the surface of the red blood cells between wild type and homozygote cells. So these proteins are enriched in DAN2 homozygotes. These proteins are depleted. And one of the proteins that was depleted was actually glycophorin A. And that surprised us. So that was also showed by flow cytometry. That surprised us because glycophorin A was present as a normal wild type gene along with all these rearrangements. So there was no reason to really believe that this was going to change the expression of glycophorin A, but it clearly did. And glycophorin A is known to be a receptor that allows parasites to invade the blood cells. So there was a logical hypothesis: if you have less glycophorin A, then potentially invasion is reduced. And so we tested that using this assay that we'd developed in my lab a couple of years before, where you label red blood cells with different dyes and then mix them all together in the same well. We call this an erythrocyte preference assay. So you add in parasites. The parasites have the opportunity to invade any of the different red blood cells, and then using flow cytometry, comparing the erythrocyte dye with the parasite dye, you can tell the proportion of parasites that have invaded the different red blood cells. And using that across a number of different strains, we could see a very clear dose-dependent effect on invasion. So these are wild-type cells, heterozygote, homozygote, and invasion was down in the heterozygote relative to the heterozygote relative to the non-Dan2, and then even further down in the homozygotes. And this was true across 5 different parasite strains taken from 5 different locations, and including one from Kenya, from East Africa, because we thought there might be some local association with the local parasites. That didn't seem to be, and strains that use a variety of different invasion pathways. And some of these parasites, like 3D7, actually they use glycophorin A for invasion, but they don't rely on it completely like other strains do, like DD2 did. So this kind of naive hypothesis that we had, that there was an association between the amount of glycophorin and the ability to invade, didn't seem to be true. And when we tested a parasite line in which EVA175, which is the ligand for glycophorin A, had been deleted, again, invasion of that strain was significantly down in Dan2 cells. So this strain cannot use glycophorin A for invasion. So if the effect was mediated by glycophorin A, there should be no effect on the strain, and yet the strain was still reduced in its ability to invade. So by this stage, we were scratching our heads and wondering if something else was going on. And this was where the collaboration with my good friend and colleague Pietro Cikuta came into hand, because he's a biophysicist and he's interested in properties, mechanical properties of cells. And using one of the assays in his lab, we looked at various biophysical properties of Dan2 red blood cells and saw something quite interesting. So the 2 key parameters here are the bending modulus and the membrane tension. And the bending modulus is essentially the force that you need to bend a cell, and the tension is essentially the force that you need to stretch a cell. And in these Dan2 cells, the tension was increased, but the bending modulus was unchanged. And concomitant with that increase in tension was a reduction in the size of the red blood cells. So Dan2 red blood cells are significantly, about 10%, 15% smaller than regular red blood cells. And actually these 2 things are linked. So an increased tension causes the decrease in size. You can think of it like the tension on the skin of a drum. It pulls the membrane in and it makes the cells smaller. So we wondered whether actually this tension could be the thing that's affecting invasion. And I remember it's one of those moments in science that doesn't come along very often, sitting, having coffee with all the key people involved at the Sanger, and just asking the question, can we measure tension, which you measure using high frame rate video microscopy, that you can see these flickers on the surface of the red blood cell that you can, through a mathematical modeling, translate into tension. Could we measure tension and invasion at the same time? And Viola Introini, who's just a fabulously talented scientist, who was then a PhD student in Pietro's lab, is now a postdoc with me, moving on to wonderful, great things soon. She's amazing. Said, I think we can do that. So that's exactly what she did. She set up an assay where, using video microscopy, first she counted the tension of all the red cells that were near a schizont that was about to rupture. Then, after the schizont ruptured, she very painstakingly counted which red cells the parasites interacted with, and then which red cells they invaded. And then she looked for the correlations between the tension and the invasion. So in this example, the parasites come out, none of the parasites contact this cell, the parasites contact these 2 cells, but they don't invade, and the parasites contact these 2 cells, but they do invade. And when she did that, she saw a very clear correlation between a successful invasion, so red blood cells where invasion occurred, and here red blood cells where invasion did not occur. So these are red blood cells where the parasites make contact, but they don't go in. And if you compare the tension of the red cells and the invasion success, there was a very clear correlation where the higher the tension, the much more decrease in success of invasion. So each of these are individual parasite-erythrocyte interactions. And it looked like there was essentially a threshold somewhere around here that if you were above this threshold, the parasites were very unlikely to invade. And if you were below this threshold, the parasites are much more likely to invade. And these are just with wild-type cells from regular donors in our lab. Then if you did this with Dan2 red blood cells, you could see that essentially, because the Dan2 red cells are shifted upwards, more of the cells fall above this tension threshold, and there's a decrease in the invasion. So our hypothesis was that essentially, if you increase the tension of red cells, you decrease invasion. And we could recapitulate the same phenomenon using very, very low doses of glutaraldehyde, 0.01%, and so on. And that subtly increased the tension, and it also decreased invasion. So you could, by modifying tension, you can decrease invasion. Why is tension reducing invasion? Well, we still don't really know, but we certainly have a hypothesis that in the process of invasion, where the parasite is essentially wrapping the red cell membrane around itself as it's driving its way in, that if the red cell tension is higher, then it's harder for the parasite to wrap that membrane around itself, and invasion is less likely to occur. And if you just try and measure the amount of membrane that's in contact between parasites and red blood cells during invasion, and you compare low tension red cells and high tension red cells, there's a very clear difference in the amount of membrane wrapping. What is it in Dan2 cells that's causing this increase in tension? We just don't know the answer yet. But if you remember back to the proteomics data I showed you, there are several surface transporters that have changed in expression levels between non-Dan2 and Dan2 cells. And we think it's potentially the case that it's the modified expression of one of those transporters that is increasing the tension. So if you use a broad spectrum transport inhibitor called Floritin, you can affect the tension of red blood cells in a significant way. And we're going on to follow that work up now. So to summarize, while going at the parasite and understanding parasite-host interactions is very important, sometimes you get clues from other places. And what we've shown is that Dan2, which is this very rare human genetic variant that provides strong protection against severe malaria, significantly reduces invasion across multiple strains, but it seems to do so not by affecting ligand and receptor interactions, or not only affecting ligand and receptor interactions. It also has another effect on the surface of red cells by increasing the tension. And so that's just a message that invasion is about more than just proteins. It's also about the biophysical properties of the red cells. And there seems to be a tension threshold for successful invasion. And we're now following this up in a number of different ways, because red cells circulate around your body for up to 3 months. And as they get older, the tension of your red cells increases. So at any one time, you have quite a range of different red cells in your body. And in fact, there's some quite good data showing that younger red blood cells are preferred by falciparum for invasion. And Viola, who did the amazing work comparing tension and invasion, is now following those up, looking at tension in these young red blood cells and seeing if it can be also could also explain why they're preferentially invaded. And the kind of take-home message is that not all red blood cells are equal, both between people, which the Dan2 variant shows, but also within people. Not all the red cells in a person are necessarily as easy to invade as each other, which I think has important implications for treatment. Just one other note at the end. I think another really important take-home message from this is it's critically important that we as scientists do more than just talk to each other and publish papers. So this project would have been completely impossible without this amazing group of people here on the left. This is the Public and Community Engagement panel and team at the Khalifi Institute in Kemri. And it's because of these amazing people that we were able to work with communities, get samples, test hypothesis. And without their work, this would have been impossible. And I think it's our fundamental responsibility to do more than just talk to each other at workshops and publish manuscripts, but also talk to the public, understand what their fears and worries are, talk to them about science, and engage with them at every opportunity. And I think the pandemic has shown that very clearly. So I'll wrap up. This is a work that was really led, most of my talk has been about Dan2. That was led by myself, Tom, and Pietro, and led by Sylvia, Viola, and Alex. But the lab, and this was just after lockdown number 2 ended in Cambridge, and we could actually go out to the pub again. The lab in Cambridge has got some really interesting new data, but because of the pandemic, it's been a bit slower than it has in the past, but we've got some exciting stuff, and they're an amazing group of people. So thank you very much, and I'm happy to take any questions.
Thank you, Julian. Very interesting data. So any questions? I suppose we have a question from Dr. Pawan Malhotra.
Julian, very nice talk. It's Pawan from
Christian.
It's Pawan Malhotra from ICGB.
Trying to unmute. Thank you, John. That was really, really interesting and very well told as well. Lovely story. But I'm just wondering, do you think it would be interesting to actually see if the parasite is able to modulate the tension of bystander cells as a way to actually promote propagation of the infection? Because it's known that bystander cells have different properties to completely naive cells.
Yeah, it's a great question. Actually, Alan has done some work showing that some of the proteins that the parasite releases do seem to modulate the biophysical properties of red cells before invasion. I think that probably our models are too simple at the moment to really understand what's going on. So with a PhD student in my lab has been trying to recapitulate this effect. We have this amazing assay, Pietro has this amazing assay where you can put parasites in contact with red cells and then pull them off again and measure the tension of that interaction, the strength of that interaction. And we've been trying to recapitulate that using beads and beads with different proteins bound to them to see if we can kind of come to some understanding. I think the reality is that we don't yet have the full complexity of the model to really understand what's going on. But I would not be surprised at all if you're right, Christian, especially if you think about the environment in the microvasculature, where there's cytoadherence, where there's probably, red cells, uninfected red cells packed around the schizont. And the chance that the parasite is not manipulating its environment, I think it's quite likely that they are, but we don't know how yet.
Yeah, that was great. Thank you.
Julia, is there any change in PTMs of cytoskeleton protein of RBC from the younger to older RBC?
That's a fantastic question. So I know that there have been some proteomic studies of progression of cells through maturation. And I also know that Manoj Joy Singh has done some surface studies to see the changes on the surface. I'm not sure that that's published yet. I don't know of any specific cytoskeleton changes, but I'm certain that it happens. So it would be interesting to compare those changes with the changes in tension as the red cells get older. But I think it's very likely that there are multiple reasons why younger cells are more easy to invade. And it could be tension, it could be, as you say, cytoskeleton changes, it could be also changes in the surface repertoire. There are probably multiple things going on.
Thank you.
It was a very nice talk, Julian. My question relates to infected RBCs. So obviously we know the reinfection of an infected RBC is difficult, and cytoskeletal rearrangements may be responsible for that. But have you noticed if infected RBCs, let's say at different stages, let's say trophozoite stages, if that has a bearing on neighborhood RBCs for fresh infection?
That's also a wonderful question. We haven't looked at that, but that's a really good idea. So we've tried to follow this tension story through the life cycle. So after the parasite invades, what happens to the red cells? But for various reasons, the longer that you image the infected cells, the more likely you are to kill them. So we haven't been able to do a full life cycle. But that idea of taking individual stages and seeing about the impact on uninfected RBCs around them, that's a really good idea. We haven't done that.
Thank you.
normal protein that aid in RBC remodeling can have role in aiding invasion of the parasites, the exported proteins. Yes.
So I think that the answer is that, as Pushkar suggested, that there's quite good data that after invasion, the infected cell is harder for other parasites to invade. So I suspect if there is a role for exported proteins, it will be in reducing invasion rather than promoting invasion. But if those proteins are exported outside of the infected cell or onto the surface of the infected cell, then as Pushkar suggested, it's possible that they may affect the tension of red cells around them. And you could also almost imagine a situation where the infected cell essentially prepares the red cells around it to make them more likely to be invaded. But that's just a model or a fantasy at this stage. We don't have any data about that.
Okay, I think these are the questions, and we thank Julian for a wonderful talk.
Thank you very much. Honored to be here.
Okay, maybe you can stop sharing, please.
So can I share my screen now?
Yes. Yes, can you just try maybe? Okay, our next speaker is Dr. Neeti Kumar. She's a senior scientist at CDRI Lucknow, and her group works on quality control processes in the cell during stress conditions. And today she's speaking on exploring unique genome and proteome maintenance pathway in the parasite, which is for alternative intervention strategies. Over to you, Neeti.
Thank you, Asif. Is my screen visible now?
Yes. Yes, can you try moving slides and you know that you are able to? Yes.
Is it moving?
Yes.
Is it moving ahead? Okay, perfect. Thank you, Asif. Thank you for the kind introduction, and good afternoon, everybody. This is the first time I would be sharing some of our data on this platform. I am a biochemist by training, biochemist, biophysicist, and my lab primarily is interested in stress biology, and we are learning important lessons of stress resilience from this enigmatic organism, that is the human malaria parasite. So we are systematically probing into different quality control machineries in the cellular compartments of this parasite. Primarily, we are looking at the protein quality control machinery or the proteostasis machinery, which comprises of protein folding and degradation machinery. In the nucleus, we are looking at non-canonical nucleic acid structures and trying to see how telomerase is helping in genome maintenance. And very recently, we have developed interest in the mitochondrion. And along with Saman's lab, we are trying to understand what would be the fission or fusion processes or the repair processes which helps in maintaining the mitochondrial homeostasis. As you all are aware that the protein quality control machinery comprises of the protein folding and the degradation machinery, and as you know that the nascent polypeptide emerging from the ribosome is protected by the chaperones at different levels. They co-translationally or post-translationally interact with the polypeptide and push the equilibrium to the folded or the biologically active state. Now, if there is any stress, they get aggregated and chaperones helps them to refold, and if these are terminally misfolded, they undergo degradation through UPS or autophagy pathway. Now, the interesting aspects is that the parasite harbors a very metastable or a aggregation prone proteome. So how is it able to maintain it in a functional state despite the environmental conditions encountered during its life cycle? So we probed into different components of protein folding and degradation machinery and did a proteome-wide analysis to see have they diverged significantly. So this is a heat map showing different components of the PQC machinery, and the color code, the gradient of blue, darker the blue color suggests higher similarity, and lighter blue or white, in fact white shows no similarity. And as you can see that many components have not only diverged from the human orthologs, but they also show significant divergence within the parasitic, closely related parasites. So we looked into the central hub of the protein folding machinery, that is the HSP 70 and 40. You might be very amazed to see that there is a unique diversification of HSP 40 proteins in comparison to the prokaryotic and human orthologs. And these HSP 40s are classified into different domains depending on the type of the presence of J domain or the peptide binding domain. And seminal work from Alexander Myers and Alan Forman's lab and others' lab in India as well as in Germany and South Africa, they have shown that HSP 40s are exported into the host RBCs, and many other HSP 40s are either cytosolic or organellar. So far, a lot of studies on HSP 70 and 40 has been done with the prokaryotic ortholog or in the yeast and the human ortholog. Systematic understanding of how HSP 70 and 40 biochemically help in protein folding of in the parasite is not understood clearly. And this was the work primarily done by Anas, and Aradhya is was also part of it, and he is currently working with the HSP 110. So our question was that, are the parasite chaperones better than the human chaperones? So we did a series of biochemical assays, and I'm just showing representative examples. We used aggregation-prone protein, thermally and chemically denatured it, and refolded it in presence of the parasite chaperones and human chaperones. And to our surprise, we found that the plasmodial HSP 70 were better in refolding the aggregation-prone protein in comparison to the human orthologs. We also did some oligomer remodeling experiments, and we used a monomer and an oligomer, and did protease protection assay or limited proteolysis experiments. So as you can see that luciferase monomer is protease sensitive, and the oligomer is resistant to protease cleavage. But when we add parasite chaperones, these oligomers become protease sensitive. Now the question is that whether there is unfolding of these oligomers or refolding of the oligomers. So if there is unfolding, luciferase activity would decrease, and if there is remodeling of these oligomers into biologically active state, then we would find an increase in luciferase activity. And this is what we found, that parasite chaperones were helping to remodel these oligomers, and they helped in increase in the activity in comparison to the human orthologs. We also did a lot of conformational dynamics experiments to see what is the inherent flexibility in these chaperones. And to our surprise, we found that although HSP 70 is conserved, but because of divergence in its peptide binding domain, the plasmodial and the human HSP 70s showed dramatic differences in their conformational dynamics, and also the HSP 40s showed. So the next experiment we did was, we asked the question, what if we swap these chaperones? So we know that the parasite chaperones are working better than the human chaperones. What if we swap these chaperones? So what we did was, we took human HSP 70 and parasite HSP 40. And in another set, we used a parasite HSP 70 and human HSP 40. And to our surprise, we found that the parasite HSP 40 was able to mediate a better HSP mediated protein folding. So this suggests that HSP 40 could efficiently cooperate with human HSP 70, thereby also giving a biochemical understanding of how the exported parasite chaperones are able to utilize the host machinery to fold their virulence factors or other proteins which are exported into the host RBC. So to understand why the plasmodial HSP 70 is HSP 40 is able to refold or mediate better protein folding, we did some binding kinetic experiments. So HSP 40 is a co-chaperone of HSP 70. So it can it actually binds to the substrates, forms this transient complex, and brings this complex to HSP 70 for protein folding. So this The forward rate of the reaction is called the K on, and the backward rate is called the K off. And the binding affinity would be the ratio of K on to K off. So we did the surface plasmon resonance experiments, and these are the sensograms showing the response unit. And we found that the HSP40 had a high of parasite had higher K on and a lower K off, thereby pushing this equilibrium to this complex formation, increasing the binding affinity of the HSP40 substrate complex. So this is how HSP plasmodial HSP40 drives the better refolding in comparison to the human ortholog. We wanted to actually take a structural biology approach, and we did an ambitious thing of planning to crystallize these proteins. And so far for prokaryotic and eukaryotic chaperones, only the domain structures have been identified, but we tried to do it with the full length protein, and because of heterogeneous conformations and concentration dependent aggregation, we were not able to continue with the crystallization experiments. So sometimes, you know, the failed experiments open new opportunities. So we took a different approach. We used these proteins for SAX based analysis, and this was Anas collaborated with Ankita from Ravi's lab, and we were able to see the structural architecture of HSP70, which is in the extended conformation, the cytosolic HSP40, which is exists as a dimer, and with the complex. This work is now being extended by Seema. Seema joined my lab in January 2020, and we are now trying to look into different cytosolic and organellar HSP40s. And these are the domain organizations of this. They have highly diverged peptide binding domains, and this also suggests that these HSP40s may have some non-canonical roles or have a HSP independent roles in the parasite. So this is just a representation of the electrostatic potential. As you can see that these proteins have really dramatic differences in their electrostatic potential, suggesting that they might have different client recognition motive. And so far, no systematic studies has been done. So our approach would be both biochemical and structural. We would like to see that what is the protein folding capacities. Is there any difference in their protein folding or aggregation inhibition capacities, and what is the difference in their client substrate recognition? So Seema has purified some of these proteins, and to our surprise, we find that they are of different molecularity. The cytosolic ones exist as a dimer, whereas the nuclear one, we were able to purify it as a tetramer, and the exported ones are either as a monomer or a dimer. So this is work in progress, and so we are systematically planning to see that how they are preventing the aggregation of a model substrate as well as for parasite specific substrates. I would now talk about another story, which also comes under the purview of protein quality control machinery. And as you know, that protein degradation is an important arm of this proteostasis machinery, and we are actually focusing on the ubiquitination pathway. So E1 and E2 are the proteins which are involved upstream, and these are relatively conserved proteins. And E3 is the E3 ligase is the protein which helps in the transfer of ubiquitin to the misfolded protein, which is then undergoes the proteasome mediated degradation. Of late, there have been evidence suggesting that ubiquitination or ubiquitin-like modifications are also playing very important role in cell cycle progression or transcriptional regulation, DNA repair, and signal transduction. So, as you know that E3 ubiquitin ligases are the one which catalyze the final step of transfer of ubiquitin to the substrate molecule, and these E3 ligases provide the substrate specificity. So among the E3 ligases, there are three major classes. One is the het E3 ligase, the other is the ring E3 ligase, and there is underrepresented category which is called the ring between ring E3 ligase. Now this is a very unique class of E3 ligase which has properties both of het and ring E3 ligase. So as you can see that it can undergo autoubiquitination as well and also transfer the ubiquitin to the target protein. So if we look at the work done in humans, there are more than 600 E3 ligases have been identified and they are suggested to be involved in different pathways. But in Plasmodium, there are very limited E3 ligases. So far, there are 3 experimental studies on E3 ligases in Plasmodium falciparum, primarily based on het and ring E3 ligase. And with the artemisinin resistance being associated with the adapter protein involved in the protein degradation pathway, these E3 ligases become even more important to understand their biology as well as to investigate their role in the parasite cell cycle progression. So Varsha is the PhD student who is working on this project, and we are working on ring between ring E3 ligase. So parasite has only one ring between ring E3 ligase, and humans have already 14 ring between ring E3 ligases, which the characteristic examples are HHARI, Parkin, HOIP. And the ring between ring E3 ligase of parasite, so this has an ortholog which is similar to HHARI. So, and it has completely diverged. It has diverged both at the N terminus, C terminus, and even the ring 1 domain also shows some non-canonical motifs, which makes it very different from the human E3 ligases. So our initial approach was to purify this protein and develop an in vitro assay so that we can screen for E3 ligases. However, despite a lot of attempts, and you are aware that parasite proteins are very difficult to purify and biochemically and structurally characterize. So we were not very successful in making a good quality protein which could be used for in vitro ubiquitination. So we took an alternative approach wherein we tried to see its polyubiquitination activity by expressing it in mammalian cells. So as you know that ubiquitin is a 76 amino acid protein and it has lysine at different positions, and depending on which lysine is involved in polyubiquitination, the fate of the protein changes. K48 mediated lysines are involved in proteasomal degradation, and K63 are suggested to participate in DNA repair. And these are the non-canonical lysine positions which have not been widely explored, and K6, K29, K33 and K27 have been suggested to play role in mitophagy, cell cycle and endocytosis. And if the E3 ligase mediates monoubiquitination, you find a single band for ubiquitin, and if it is a polyubiquitination, you find the smear. So this was a strategy. We ectopically expressed the parasite E3 ligase with a HA tag ubiquitin in mammalian cells and did IP, and this is how we get the ubiquitin smear. The next thing that we wanted to see that whether this E3 ligase mediates K48 or K63 linked polyubiquitinations. For that we used ubiquitin in which the lysines were mutated, and we had lysine only at K48 or K63 position. And when we did IP, we found that yes, it is able to mediate both K48 and K63 linked polyubiquitination, suggesting that it may play role in proteasomal degradation and in DNA repair pathways. Then we try to map the critical sites which are important for ubiquitination, and we try to we tried to filter out few positions, and in ring 1 and ring 2 domain, actually the E2 enzyme interacts with the ring 1 domain and then transfers the ubiquitin to the ring 2 domain. So these were the mutations that we investigated, and we found that both mutation, point mutation in ring 1 or in ring 2, we abrogated its polyubiquitination activity, suggesting that these positions are very, very critical for the autoubiquitination activity of E3 ligase. In parallel, we tried to use yeast as a model to identify which could be the probable pathways this E3 ligase is participating in it. So the yeast homologue of parasite RBR-E3 ligase is a HEL1, which is a histone E3 ligase. So we shortlisted two histone E3 ligases, and we subjected them to different proteotoxic stress conditions, both hydroxyurea. We also gave MMS and other stress conditions to see whether there is a rescue in the phenotype because of this stress. And to our surprise, we found that E3 ligase was not rescuing any of these phenotypes, which may be because the parasite ligase would have evolved specific species-specific functions, and that is the reason why it was not able to complement these deletion strains and rescue them from the stress. We also tried to identify the corresponding E2 enzymes of RBR-E3 ligase, and this they were the homology model suggests that they are similar to UBC5 and UBC13, and through these two hybrid, we showed that yes, indeed, these E2s are interacting with the parasite E3 ligase. We tried to purify these proteins and raise polyclonal antibodies, and when we purify both these E2s, they are coming at 16 kilodalton, but when we are checking the antibody in the lysate, we find higher molecular bands being detected, so probably suggesting that E2s in the parasite E2s are forming higher order complexes. And for the E3 ligase, we were not able to get a very single band as Sumandhar and Saman had also shown in their presentation, that sometimes when the proteins are getting processed or post-translationally modified, we do not get a single band for these parasite proteins, and this smear that we see here probably could be suggestive that it is either getting processed and also post-translationally modified. We did some localization experiments and found that yeah, this ligase is present both in cytosol and nucleus, and we subjected the parasite to different proteotoxic stress conditions, and we found that specifically when we give DNA damage and the mitochondrial stress, so these are Giemsa stains parasite showing pycnotic appearances, and this is the untreated control, and we found that there is a specific increase in expression of our E3 ligase, the plasmodial E3 ligase. We also subjected the parasite to Mg132, which is a proteasomal inhibitor, as well as to heat shock, but we do not see any increase in the expression level. Since these experiments are indicated or suggestive of that they might have role in DNA damage or in mitochondrial damage, we are actually now doing some experiments to see whether E3 ligase is involved in mitochondrial homeostasis, and we are trying to see whether it is mediating K6 linked ubiquitination. That would give us a hint whether this E3 ligase controls the fate of proteins through K6 mediated ubiquitination, which has important role in maintaining the mitochondrial homeostasis. I will quickly switch gears and talk about genome maintenance, and as you are aware that the parasite nucleus, especially Plasmodium falciparum. It has a 80-rich genome, and the telomeric and the subtelomeric regions only are the regions which harbor GC richness. And you know that telomeres are very important for maintaining the length, and decrease in telomere length can lead to cell death. And telomerase is the enzyme which maintains the telomeric length. So unlike humans, where telomerase is only expressed in cancerous condition or under in certain germ cell types, parasite telomerase is expressed in all the stages. Now, if we want to perturb the telomere dynamics, we can use the strategy of targeting these telomeric ends which have G richness and they have the propensity to form secondary structures. So using quadruplex interacting ligands, if we push the equilibrium to these non-canonical secondary structures, we may interfere in the telomere maintenance by telomerase. And this is possible because there are dramatic differences in the telomere dynamics both in host and the parasite. Now, the telomere in the Plasmodium falciparum is 1 to 2 kb long, whereas in humans it is 10 to 15 kb long. And telomerase is 2.5 times bigger than human telomerase. And the proteins which bind to the telomeric and subtelomeric region are highly diverged, and we currently do not have comprehensive understanding of this. So, for the proof of concept studies, we had used quadruplex interacting ligands. We got these compounds from Pradeep's lab from IIT Bombay, and we checked it for parasite telomeric DNA, and we found that yes, the telomeric DNA of parasite can form quadruplex, and in presence of these compounds, there is a conformational change in these structures. And when we treated these compounds to the parasite, we found that there is a decrease in the telomeric smear. We extended this study with other quadruplex interacting compounds, and as you can see that not all the quadruplex interacting compounds show good activities. We stumbled across this furyl benzamide compound, and we found that it had significant activity, 65 nanomolar, both in sensitive and good activity in the chloroquine resistant strains, and with the CC50 of cytotoxicity of 30 micromolar. Now, if this chain is extended, then this compromises the activity of antiplasmodial activity. So, to understand what is the rate of kill of this molecule, we did this quick experiment wherein we treat the parasite at ring stage at lower doses for 6 hours, and then after drug pressure removal, we let the parasite grow and then do a FACS analysis. It is similar to what the previous speaker had suggested for mixing of dyes for infected RBCs and uninfected RBCs. So we did FACS experiments, and the Q2 quadrant would suggest, would be indicative of viable cells, that is, which is positive for mito-red and positive for cyber-green, whereas the pycnotic cells would be mito-red negative. So what I want to say is that slightest perturbation in the subtelomeric and telomeric region, how is it actually transmitted? That communication is transmitted to the mitochondria, we do not completely understand, and this is what we are planning to do. This is the work. Nirupa is the student. She joined my lab in November 2020, and we are actually now probing into what is the communication from the nucleus to the mitochondria, and is parasite telomerase playing important role, because what we know from the mammalian system is the human telomerase does get translocated into the mitochondria. So the experiments we are planning to do, Nirupa is raising antibodies against different domains of the parasite telomerase, so that we can check the localization and use it for CHIP to understand the regulatory roles of telomerase in telomeric and non-telomeric regions. And also she's trying to establish the telomerase assay, so that we can screen for compounds which show which can inhibit or which can disturb the telomere dynamics. And this brings me to the end of the presentation. This is my current group, and I'm very proud to mention that Aradhya and Anas were the first batch of volunteers, batch of student volunteers who helped in setting up the COVID testing lab in CDRI in April 2020. And during the same time, our lab, along with Atul's and Ashish's lab, we ventured into a new area. Because we were, as you all are aware, that there were questions about how to import fluorescent dyes and quenchers for Tachman probes for RT-PCR based diagnosis of SARS-CoV-2. So Atul is a medicinal chemist and Ashish is structural biologist, and so we all teamed up and we prepared in-house fluorescent dyes and quenchers. We did labeling of the dyes and tested these Tachman-like probes in the COVID positive samples that we got from a CDRI facility, and this technology was transferred to biotech test. And of course, all the fundamental and anti-malarial work cannot be done by without blood donors and the volunteers, so they are acknowledged, and we closely interact with the malaria, the structural biology, and the medicinal chemistry group in CDRI. These are the collaborators, and thank you for your attention. I would be happy to take questions.
Okay. Thank you, Niti. Thank you very much. So we have one question from Dr. Dinesh.
Yeah. Nice talk, Niti.
Thank
Can you hear me? Yeah. So my question to you is, have you checked that similarity between human turd and PF turd?
Yeah, but it is only some catalytic residues are conserved, but the entire protein is very, very different. It is 2.5 times bigger than the human telomerase, and in fact, the telomerase RNA is 3 times bigger in Plasmodium in comparison to the human one. So it's very, very diverged.
And second question is that many TERT inhibitors are already known for human. So are you planning to try some of them on Plasmodium as well?
Yes, yes. So that is the plan, and we would like to prepare parasite-specific telomerase assay, and this is what Nirupa is trying to standardize now. That if we can develop in-house parasite-specific telomerase assay, then we can compare whether the available inhibitors, how are they working with human telomerase as well as with parasite telomerase.
All right, thanks much.
Thank you. Just one question from my side. So for these ubiquitin ligases, you have this cell-based assays. So how good are these assays? Can you screen some compounds using those assays? Because I know Matthew Boggio's group has developed a number of compounds for E3 ligases. So I thought if you can check, you will know specificity.
Yes, so actually we wanted to do a in vitro assay wherein you add purified E1, E2s and E3s and the compounds, but we were not able to develop a in vitro assay, a quick in vitro assay. So that is why we had to go through this transfection in mammalian cells to identify. But yes, using this indirect approach, by seeing ubiquitination, diminishing of ubiquitination smear, we can differentiate whether it is specifically working on the parasite E3 ligase or is it working with the human E3 ligase.
Okay, so I see no other questions. So in that case, we will thank you for your wonderful talk. Thank you very much for joining.
Thank you. Thanks a lot.
So maybe you can stop sharing this.
Can you hear me?
Yes, Cyril, I'm just giving you the control. Stop preview, it is already there, I think it's okay. Okay, Cyril, you can try to display your slides.
Okay, give me a second. I'm having a little bit of trouble. I don't know why. Share screen, please.
You can see your screen.
Yes.
Yes, now we can see your screen. Can you put it on display mode and move slides?
Seems to be working.
Yeah. Okay. So our next speaker is Professor Cyril Botte. He is from University of Grenoble in France, and he has done pioneering work on lipidomics, especially linked with the organelle and which is linked with our dynamics. And his talk is, it's all about lipid.
Thank you very much, Asif, for this nice introduction, and it's good to see you, especially with the COVID times. I would like to thank all of the organizer at ICGB for this very nice invitation, for such a nice workshop, and for giving us the opportunity to talk about what we're trying to do and trying to understand how epicomplex parasites acquire their lipids and synthesize them, and how they sustain parasite survival at different levels. Okay, so I'll quickly jump to the introduction. We all have heard about epicomplexan parasite, the unicellular eukaryotes responsible for major human disease. You've heard a lot about malaria. You may have heard a little less about toxo, although Pushkar has been talking about it. So we know that there is no fully efficient vaccine, although there's been the authorization of the RTSS by the WHO. And also there's the problem of multi-resistance to existing drugs, especially for Plasmodium. This I will skip. So they are obligate intracellular parasite. They have to invade a host cell in order to propagate and survive. And one of their peculiarity is that the epicomplexan harbor a non-photosynthetic plastid, which is named the apicoplast, and this plastid has been acquired by the secondary endosymbiosis of a red algae. Although the apicoplast has lost its photosynthetic capacities, it is metabolically similar to plant and algal chloroplast, and it is essential for parasite survival, making it an interesting drug target as it is solely present in the parasite and absent from the host. All right, so what is the link between apicoplast, lipid synthesis, and parasite propagation? So throughout the intracellular division cycle, parasites require lots of lipid to generate the membranes of the future daughter cell for signaling and for storage. And for a long time, if we schematize things like this, with here being the host and here being the inside of the parasite and here being the parasite first vacuole, we don't thought that parasites were solely relying on the host lipids to generate their own membrane compartments. However, the discovery in the late 90s of the apicoplast, and most importantly of its prokaryotic type 2 fatty acid synthesis pathway, the FAST2 pathway, suggested that the parasite was capable of de novo synthesis of fatty acids, which are the major building block, hydrophobic building block for membrane lipids. On top of that, the FAST2 pathway depends on the scavenging of host glucose, which is transformed by parasite glycolysis to provide for the substrate of the FAST2 pathway to make fatty acids, and I'll get back onto this. Oops. The initial characterization of the FAST2 pathway and its knockout showed that the pathway is essential for parasite survival, especially during a very active division cycle, such as during the acute phase of toxoplasmosis during tachyzoid life stage. As you can see here, when you knock out the FAST2, the parasite collapse and there's normal division, as well as during the liver stage of the parasite, where it blocks parasite biogenesis, and also during schizogony in the mosquito at the sporozoite schizogony level. And it seemed that the FAST2 was not essential during blood stage, but I'll get back onto this. On top of the FAST2 pathway in the epicoplast also harbor two acyl transferases homolog to the plant chloroplast enzyme named ATS1 and ATS2, which are sequentially capable of esterifying a fatty acid onto a glycerol 3-phosphate backbone in a two-step pathway, forming lysophosphatidic acid and then phosphatidic acid. And phosphatidic acid is actually the sole precursor for the de novo synthesis of all phospholipids, which are the main membrane lipids found in biological membranes. So the apicoplast seems theoretically capable of making the building blocks of lipids and synthesizing major precursor that was based on gene presence. So at the time we started the studies, we didn't know what was the contribution of the actual de novo synthesis pathway happening in the apical plus, but also in the ER, and also what is the exact contribution of the scavenging pathway, and what is the nature and the destination of these lipids, how they're trafficked, and how they go across several membranes. And these are the questions that we are currently asking. So the first question that we had is the apicoplast Fas2 actually always on, meaning essential, or off, completely dispensable throughout the parasite life cycle? And to answer this question, we use stable isotope labeling combined to lipidomic analysis. Very briefly, as I said, the parasite is capable of using glucose to form acetyl-CoA, which is the substrate for the Fas2 pathway. And this acetyl-CoA is fused to malonyl-CoA, 2 carbon plus 3 carbons, to form a small chain of fatty acid, which is the addition of carbons. And throughout each cycle, you have the addition of 2 more carbons. And if we depict the glucose as with its carbon, it's 6 balls like this. And if you look at lipidomic analysis, mass spectrometry, the mass of a typical fatty acid of 14 carbons is seen as a mass of 242, or the mass ratio seen with the machine. And this is how it's seen. Now, to monitor the activity of the pathway, we use stabilized isotopically labeled glucose, where all of the carbons, 12 of the glucose, were replaced by the stable isotope C13, which have a mass difference of +1 per carbons. And these are the red balls seen here. So if you schematize this, again, the glycolysis can form acetyl-CoA and then malonyl-CoA, which can be used for the Fas2 pathway to form C14-0. And theoretically, you have the form of unlabeled fatty acids in here with this mass of 242 for 14 carbons. But when they're labeled, well, this is what we see, a mass at 256. And if you make the difference, 256 minus 242 equals 14. So you can then quantify the amount of carbon that have been incorporated in the chain. And that's the principle of how we measure the activity of the Fas2 pathway. This protocol has been set up with Professor Malcolm McConville at the University of Melbourne, who we're working together. So now, to understand the contribution of the Fas2 pathway, so we used that, and we fed either C12 glucose or C13 glucose to see the formation of fatty acid by the parasite. And we did that in blood stage, sexual blood stages. And what we could see is that when we cultured the parasite in the regular culture medium, which is rich either in serum or in albumax, which are not very physiological conditions, but rather very rich conditions, then what we could see is that we had an abundance of unlabeled fatty acid chain, meaning that the Fas2 pathway was not active. However, when we change the nutrient availability, the disponibility of nutrients and lipids in the medium using the control the control culture medium set up by a Japanese group a few years back, Mitamura et al., then what we could see is that when we deprive the parasite from nutrients, we see an incorporation of carbons by the Fas2 pathway. The pathway becomes active, which is different from what we thought, that the pathway is actually dispensable during blood stage. It's not, it's rather more back into a physiological conditions where the pathway can be turned on and off depending on what's available in terms of nutrients. And we went a little further here. We used the Fas2 knockout, the Fab-I knockout, which is here in green and purple, versus the wild type in blue and red, put it in a rich medium or starved medium. When you have the Fas2 pathway in blue and red, in rich or in starved, you have growth anyway. Where if you knock out the Fas2 pathway in rich medium, it grows, but in starved condition, then the parasite struggle to divide and then eventually dies or at least disappear from the culture, meaning that the Fas2 pathway becomes essential when the parasite needs it during blood stages. And this was also quantified and validated in Toxoplasma gondii, where when you change the nutritional conditions, then the Fas2 pathway can be upregulated. Another thing that we've seen in Toxoplasma gondii during lipid-deprived conditions, so if you see here a vacuole of parasites within a host cell, what we could see in lipid-deprived conditions is that we saw the accumulation of these giant multivesicular bodies containing vesicles of different sorts, including lipid droplets as well as membrane-bound vesicles that were accumulating next to the PV, and then their content percolating, being actively imported by the parasite. And if we looked in details, these giant multivesicular bodies were arising from the host organelles, mostly from the nuclear envelope here, as well as from the ER and the mitochondria. So our studies, basically briefly, the take-home message is that when you have the parasite in a normal or rich environment, then the parasite can uptake lipids from the external medium, from the host cell via lipid droplets or other organelles, and the apicoplast fast 2 can contribute depending at a low basal level or a higher level, depending in toxo or in plasmodium. However, when you change the nutritional condition, there's a change in the parasite metabolic program, basically, that upregulates its capacities to make fatty acid via the FAST2 pathway to boost the amount of lipids, but also there's a deep cellular change in the host cell where the parasite sends effector to have these giant multivesicular bodies forming to have more membrane material. This is at least happening in Toxoplasma. And in Plasmodium, it's a little different because, of course, you can't find all of these organelles, but there are other things happening. So that puts back the parasite into more of a physiological state where some genes can be essential depending on what is happening outside. The parasite can sense and use what is necessary for its own survival, rather than black or white or a binary system where you have a pathway that is thought to be always essential or dispensable. And these findings were correlated by other findings that also inspired or proved us right. For example, that different patients with different nutritional states were having different transcriptional patterns where the FAST2 pathway was upregulated in terms when the patients were put in starvation, or when in Plasmodium berghia, when you reduce the diet and you reduce the number of progeny, the merozoites, or when the lipid precursors, main lipid precursors such as lyso-PC, are lacking to make more membrane during the parasite during asexual blood stage, then the parasite shift into slow dividing gametocytogenesis. All right. Now, going back to the capacity of the apicoplast to use these fatty acids. So I remind you, we identified acyltransferase called ATS1 and 2 that could be virtually capable of making lysophosphatidic acid and phosphatidic acid. To understand their roles, then we started looking at the protein ATS1 here. And then so we tagged it in both Plasmodium and Toxoplasma. This was done by previous PhD student Melanie Shears, which was co-supervised with Jeff McFadden and I at the University of Melbourne, and Suad Amir for Toxo on the French side. So when they tagged the protein, they found it in the apicoplast of both Plasmodium and Toxo. It's not shown here, but you have to believe me. And this is what's happening during the liver stage. So you see that when the ATS1 is valid, then you have the growth of apicoplast and multiple nuclei and multiple plasma membrane, I see here, for the future merozoites to be. But when you shut down the enzyme, then you stop having the formation of the membrane of the future merozoites, and then the parasite stops dividing, there's much less nuclei, and the biogenesis of the organelle is completely stopped. Similarly, in Toxo, so here you have a Toxo with its parasitophorous vacuole, and here a plaque-assay, and the white areas shows where the Toxo is lysing the host cell. This is also what's used for COVID to measure the growth of the parasites. When you shut down the enzyme, basically you have an arrest of the intracellular development, as well as an arrest or something wrong with the biogenesis of the intracellular compartment. So to understand the role of ATS1 for these processes, we, and especially Yoshiki, our head of our lipidomic platform on site, or Gemili, has used the approach that we use with using C13 glucose, where we would be feeding either unlabeled or labeled glucose to monitor the synthesis of labeled fatty acids. And here, ATS1 would be placed virtually here to potentially incorporate fatty acids to form lysophosphatidic acid. And so we measured this by mass spectrometry approaches. And what we found is that the parasite was making labeled lysophosphatidic acid specific of lengths 12,0 and 14,0, short chain fatty acid, which are the signature of faster synthetic capacities. And these fatty acids were incorporated into lysophosphatidic acid. And these lysophosphatidic acid was used as the backbone for all other phospholipids. And we confirmed this by complementing knockout in toxo with the product LPA14-0, and led us to this actual model where the parasite is capable of taking the host glucose, form acetyl-CoA via S-glycolytic pathway, import PEP into the epicoplasm, make fatty acids, and these fatty acids can be incorporated by ATS1 to form lysophosphatidic acid. This lysophosphatidic acid is then exported and combined to host fatty acids to make the bulk of toxoplasma phospholipids. Without ATS1, you lose about 50% of the full mass of phospholipids, and you lose all of the epicoplasm origin fatty acids. That also leads to say that the parasite has to make patchwork lipid, where it takes fatty acid from then it makes itself and combine it from scavenged fatty acid. And without this combination, at least in toxo during acute stages and probably in liver stage, you need these patchwork lipids. All right, so we looked at ATS1 and we know the parasite can make this. How about the phosphatidic acid? What is its fate and what is it happening? We wondered this, and we looked at also now the scavenging pathway. So we looked at the enzymes that were capable of utilizing phosphatidic acid to form diacylglycerol, because diacylglycerol is another central precursor for phospholipid synthesis as well as for storage. And the parasite possesses several phosphatidic acid phosphatase, PAP, and one subfamily that's called the lipin, a single one in each Toxoplasma and Plasmodium falciparum. So we decided to look at this. And this was the work of Sheena Das, a previous PhD student who was working with Sumit Rathore at ICGB and AIMS, and also Serena Shumungam, which is a sandwich PhD student with a Cefipra project between ICGB with Asif and us. So when we looked, we tagged the protein, and we found that it was cytosolic, avoiding the nucleus area, and most likely also colocalizing with endoplasmic reticulum markers. So we made an inducible knockout by changing the promoter to a TET-off system, which is the typical ways of making mutants in toxo, although the AID system has now been taken over. And then we confirmed the loss of the protein after 24 hours and 48 hours complete absence of the protein, confirmed by IFAs where the signal was lost. And so in the mutant, what we could see is that we had a significant reduction of large vacuole with lots of parasites, significant increase of smaller vacuole of 1 or 2 parasites, and a lot of abnormal vacuoles with parasites that were dying. And so we did plaque assays, and you could tell that the protein is very essential. However, there's a little twist here. So when we, knowing that from our previous results that the nutritional environment is important for the parasite, we changed it to 10% FBS, high nutrient, 1% about normal, and low starvation. When the parasites tend to grow better in higher nutrient environment, here in the mutant, it was the other way around. The more nutrient we were putting in the parasite, the least the parasite were able to grow. So there's something wrong with having more nutrient and no lipin. It kills the parasite. Now, what is the phenotype of those parasites? So what we could see is that the parasites were having, so here are the mutants at different 0, 1, and 10, so starved, normal, and high nutrient. And this is what the parasite look like. So they look with like evaginations like this. then they start to be distorted and fused to stop division at high nutrient. So again, phenotype was being confirmed at higher nutrient. You have more phenotype in the parasite, can't divide and dies. Even we could see the accumulation of endoplasmic reticulum whorls, or big chunks of membrane accumulating in the parasite without the lipin, even at normal nutrient conditions. So there was something wrong with the membrane biogenesis of the parasite. So we decided to look at what was going on at the lipid level. And so we separated the lipids in the main classes: PL phospholipids, so the membrane lipids. DAG and TAG are, so DAG is a precursor, as I said. The TAG is triacylglycerol, storage lipids. And free fatty acids are the building blocks for either making phospholipid or a storage lipid. Now, what we could see is that we had a significant increase of the accumulation of free fatty acids and a loss of the storage lipids that was going on in the parasite. So we were not very expecting this at the storage level. And this was confirmed by looking at the lipid droplet of the parasite. So the lipid droplets are these organelles that are storing triacylglycerol, diacylglycerol, and neutral lipids that the parasite or any eukaryotic cell can use when necessary. And we could see that there was a significant decrease of lipid droplet that was dependent on the level of nutrient present in the parasite, and so was TAG. So we're wondering, so what is, so we had this accumulation of free fatty acids and a decrease of storage lipids. So where is the accumulation of free fatty acid coming from, and where do we have a loss of TAG? To do so, we pushed our stable isotope labeling a little further to monitor the different origin of lipids. So I already presented you the fact of using stable isotope labeling with C13 glucose to monitor the apicoplast FAST2 activity. And then we use another technique. We pre-label the host cell with C13 glucose without the parasite. So we let the host cell making its own lipids. So all lipids are labeled. And then we put the parasite, and then we let the life cycle go and take the parasite, and any lipid that is labeled is actually from the host cell, so we were able to quantify the scavengome, what is the parasite taking up. And we even did that in the external environment. In the medium, we used labeled fatty acid, in this time deuterated fatty acid, because C13 carbon labeled fatty acids are a little more expensive. And so we're able to quantify things that were coming from the outside, from the host cell or from the parasite, to see and explain our phenotypes. So first, we looked at the FAST2 pathway. And so we looked at the FAST2 activity in all of our fatty acids, and what we could see in the absence of so the mutant is in orange and the wild type is in gray. In the absence of the lipin, we have a decrease of the FAST2 activity. We have a decrease of the free fatty acid present in the parasite, and we have a decrease of the phospholipid coming from the FAST2 pathway. So first answer, the free fatty acid accumulation in the lipin mutant doesn't come from FAST2, and it seems that in the absence of lipin, FAST2 is decreasing its activity, as we previously seen with the metabolic reprogramming. Then we probed at the external environment by putting fatty acid in the external environment, as I said, and again, total lipids lower, free fatty acid lower, and host-derived fatty acids about stable, even lower. So this is not lipids from the external environment that lipin is taking, is using. But then, when we probed the lipids made by the host cell, what we could see is that in the mutant, there was significant increases of fatty acid made by the host cell, especially C18-1, which is very important for the lipid droplet accumulation. And we saw a concomitant decrease of these fatty acid made by the host cell, especially C18-1. So that meant that lipin is using the flux, the constant flux of fatty acids arriving from recycling the host phospholipids. So that leads us to this model. In normal growth condition, the parasite uptake lipids from the outside, especially from the membrane of the host cell. And it's made into phosphatidic acid, and then the lipin makes diacylglycerol from it. And this diacylglycerol is actually used to accumulate in triacylglycerol, which is then mobilized for membrane biogenesis when necessary. And another part is coming from the fast food pathway. However, when you get rid of the lipin, what is happening is that you mess up with the whole program of balance between pathways and how the parasite can survive. So first, if you inactivate lipin, what is going on is like first the parasite can sense it and lowers the amount of fatty acid made by the fast food pathway, but there's no more DAG and there's less DAG and less lipid storage. So the parasite thinks that it needs to scavenge more. So there's more fatty acid scavenged from the outside, and these fatty acids can't be used by the lipin. So they accumulate as free fatty acid, as PA, and there's lipotoxicity. And so the parasites try to cope by making more membranes, which is a resulting world of membrane of ER happening. And even more in toxo, we saw that even not in starved condition, there was this giant multivesicular body, even having more lipids. So the parasite basically dies of obesity, not being capable of fluxing, channeling the toxic flux of fatty acids coming from the host as a constant flux, because the parasite actually constantly scavenge the external environment. So that leads us to this model where both host lipids and apicoplaslipids, at least in certain time points, are essential for making patchwork lipids and lipids of the parasite. And these patchworks are extremely important in specific life stages. But we also saw that the host nutritional status has a direct influence on how each of these pathways works and help the parasite acquiring lipid and remodeling to make and maintain the lipid homeostasis in the parasite. The current question is, how is the parasite mixing these different fatty acid sources to make this patchwork lipids? So basically, what are the enzymes involved in this? And how does the parasite sense the host to change its metabolic program and use the proper pathway when necessary? So these are the things that we are currently on, and there are several enzymes that could be involved in there. One such type is lysophospholipase, and I'll have a word about it. Let me see how late I am. Seems to be okay. And Asif, tell me if I'm running late. So you have phospholipases. Phospholipases are enzymes that are capable, virtually capable of cutting fatty acids up. 2 minutes or something, I'll be good. Perfect. Perfect. So lysophospholipase are capable of cutting fatty acids from existing lipids. So either made inside and being reshuffled, or taken from the outside, because this is what we see is probably happening. These fatty acids need to be activated, transported, and there's a whole lot of transporters and activators that were also interesting. But I'll give a word on phospholipases. If you look in falciparum at the number of phospholipases and lysophospholipase, you can see that there are many more than regularly found in eukaryotic cells. And on top of that, in falciparum, these phospholipases are present in the subtelomeric regions, where we usually find the VAR genes, rifin, and other markers of phase that goes under a lot of recombination to have a more diverse family. Actually, the phospholipases and other metabolic genes dealing with fatty acids and lipid metabolism are located there, meaning that maybe the parasite need this function and need to have redundant function to maintain in order to maintain parasite survival. And I'll give you 2 examples that we've studied with Steve throughout our CEFIPRA collaborative project. One of these lysophospholipase called LPL1 is found in vesicle traveling from the periphery of the parasite towards a compartment next to the food vacuole, as seen here in immuno-EM, accumulating next to it in a neutral lipid rich environment. And when we looked at the lipid profile of this mutant, we saw that there's an increase in the mutant of fatty acids, meaning that the fatty acid made by generated by the enzyme, as seen here, are increasing. And then we have an increase of DAG and a decrease of TAG, meaning that this fatty acid could be used to make TAG. And most importantly, the level of hemozoin is completely reduced in the mutant. And basically, to make a long story short, because I don't have much time, actually the lysophospholipase provides fatty acid for the synthesis of triacylglycerol, which is used as a start point for hemozoin polymerization and help the polymerization of hemozoin. So without these lipids taken from the outside and used for triacylglycerol, the parasite is dying. And this was also confirmed by the addition of external fatty acid sources in the mutant to confirm what I was saying. And another lysophospholipase that has a different role is found in the peripherin, in the first vacuole, the first vacuole, sorry, as well as the intravacuolar network that is in the red blood cell. And when we knock out this enzyme, we have a significant reduction of progeny. And when we look at the lipid levels, we have again the TAG and DAG ratio that is affected, and the parasite starts having less TAG, less DAG, more free fatty acid and more phospholipids, which was confirmed by fluorescent analysis and accumulation of fluorescent fatty acids in the absence of the enzyme. That leads us to this model, very similar to toxo, that the parasite in normal conditions take lysophospholipids to create fatty acid, to generate fatty acid as well as other polar heads that are used. But these fatty acids are used to generate DAG and TAG that are stored in lipid droplets and used when necessary only during schizogony to mobilize them. In the absence of it, then you have a decrease of TAG, decrease of DAG, and normal membrane biogenesis when necessary. And an important point, especially for the workshop of today, this LPL3 has been found to be targetable by non-MMB compound, as shown here, in the low micromolar and high nanomolar level, and it blocks basically the division of the parasite that can't divide. And we did lipidomic analysis showing that they were phenocopying the lack of the enzyme. So these are very good compound candidates to actually block this, to understand the lipid metabolism, but also as potential drug as for therapeutical intervention. On this, I'll stop. I hope that I've convinced you there's a lot of process that depends on lipids that we didn't know of, and how the parasite is doing it is very, very complex. I would like to thank all of the people in our team, especially all of the PhD students and the previous PhD students that did the work, especially Sheena, Suad, Melanie, Serena, Nick, everybody. Our collaborator, Jeff McFadden, Malcolm McConville, Asif, and Pushkar. Pushkar presented the story yesterday, so I did right in not presenting it again. All of our funders, CNRS, Fondation pour la Recherche Médicale, and etc. And also thank our capacities to perform lipidomic and fluxomics at our own platform here in Grenoble. And if you have a lipid question and you want to work with us, don't hesitate to contact us. All right, I'll stop here, and I'll thank you for your attention, and I'll be happy to take on questions.
Thank you, Cyril. So we have a question from Pushkar and then a question from a student. Pushkar, please go ahead.
So nice talk, Cyril.
Thank you.
Yeah. So the question I had was, so when you have low nutrient levels of low fatty acid in the extracellular milieu, in case of Plasmodium and maybe also in Toxo, where do you find, is there a trend in lipid biogenesis which is apicoplast driven versus ER driven, or is it, or there is no such trend?
So when you shut down one way or the other. Say in your question, it's when you were in low nutrient, do we have a trend of more ER or apicoplast? Yeah. We have more de novo synthesis pathway happening, both at the FAST2 pathway, but also in the ER. You know about the acyl transferase that we've been working on with the GPAT, and it seems that in toxo, I've seen other people that have shut down the apicoplast, and so you lose sort of the FAST2 pathway for other reasons, indirect reasons, and we can see that the ER is taking over by fluxing more from the outside and having these guys working more in trying to compensate. So there is this thing of de novo scavenging. I take over, I shut down because I'm lazy and I don't need to use energy, so I'll take more from the outside.
So my final question, I mean, all of this, how are these fatty acids taken up in the case of, let us say, Plasmodium? I mean, of course, they could be, LDL receptor mediated, this thing, but probably that's not the only thing. I mean, is it the TVMs or there are multiple ways? I mean, how are these fatty acids taken up?
So knowing my parasites now, I would say that it's multiple and complex, and you have partly mentioned some of the things that is happening. So in toxo, I would say sometimes there's like a whole bunch of big vesicles and even big lipid droplets that seems to be passing through the PV, and Isabelle Coppens has been showing this. Also, we have direct contact sites with some organelles, and so we have a flux of lipids this way. And some Chinese groups showed that by ACBP2 in toxo, there's like a flux from the mitochondria, although Lina Pernas also showed that there's a battle there. And there is the recycling of existing lipids that is happening. So the parasite takes phospholipids or existing lipids and cut it into pieces and reassemble it. And that's the most intriguing part. Why does it not take it as such and use it as such? It needs to cut it in pieces and reshuffle it. There are particular reasons. I've told you about the patchwork. There's the size of the fatty acids that are important and everything. And to finish answering your question, not only the membranes of the host cell is contributing, but also from the outside directly. And when you ask me of how this is happening, well, let's work together and find some more transporters. There are plenty of candidates, and I think they all have a specific role. When you see this LPL1 versus 3, one is promoting hemozoin polymerization, whereas the other one is more for progeny making. There's like many things that the parasite is meeting together to obtain what is central, at least to me, the lipids.
The only problem with talking to you is you start making you think about lipids, where, something which I had been running all away from my life. I mean, all my life I've been running away, and as luck would have it, you get entangled with that. So anyway, thanks very much, and thanks, Asif, for allowing me.
Well, thank you, Pushkar. And the lipids are not that bad. We even published together on lipids.
But they need experts like you.
Thank you.
Yes. So there is a question from PhD student Mujahid. So they said FAST2 is activated in gametocyte. So is it their inability to uptake host lipids and they have constituted FAST2?
That's a very, very good question. I hope that you asked it to Alex Meyer a little earlier as well. This is clearly a possibility, and to be honest with you, I don't know what's the name of the student, but that's fine. I'll find out in a second, I guess. But there's a thing that hasn't been tested. Can gametocyte uptake things from the outside? And that's something that needs to be tested. Because, of course, your point is right. With what I said, there's like a compensation between scavenging versus de novo. And it seems that if one is turned on, usually that's because the other one is not available. And it's a very, very good hypothesis that has not been tested. But what I can tell you is that maybe that's the two, because if you see in liver stage, scavenging is massive, and FAST2 is very active, although the parasite is in the factory for making lipids. Why does it still conserve both pathways, especially when it's thriving in an environment that's thriving of lipids? So yes, it could be because the gametocyte can't import anymore, but it could be also for something else. Something that I haven't told you is that one of the acyltransferases of the epicoplast provides some phosphatidic acid and lysophosphatidic acid to create local membrane curvature for having dynamine to insert at special pinching points. And maybe there's such a thing happening. I'm not saying that gametocyte needs pinching point or dynamine to divide, but maybe there is something very specific in terms of the product, because we have to remember that fatty acids providing shorter fatty acids.
Okay, another question from a PhD student, Shilpi Jain. So she's asking, in lipid knockout, is there upregulation of some transporters or proteins which are responsible for lipid uptake?
Yes, yes, I can't be very more specific in terms of what and who. I'm happy to talk this more particularly, but yes, there are some transporters being upregulated. And as I said, and it was a question that you, Pushkar, also mentioned, when there is something shut down, the parasite has to compensate, and there are usual suspects. For example, some of the triacylglycerol acyltransferases or some of the lysophospholipases that are easy provider. But yes, there's always it seems to be always a compensation.
Okay. I think there is no other question. And in that case, we will thank you, Sayal, for a wonderful talk and for joining us. And hopefully next time you will join us here physically. Thank you very much.
My pleasure. I hope to join you very soon and enjoy seeing you again, or vice versa. Have a great meeting. Thank you. It's fantastic.
Question? Oh, there's a question again. Oh, sorry.
Oh yeah, go for it.
Question. I can open here. So there is a. Oh, sorry. The question from Mayuri.
Yeah, I'm reading it. I'm reading it.
Oops. Has three pathways for fatty acids: fast 1, fast 2, and the scavenging. According to you, what is the role of fast 1 pathway? And have you conducted any experiment to confirm the same?
So it's a great question. Thank you for asking, Mayuri. So if you look more carefully, there's even more. There's 4: fast 1, fast 2, scavenging, and even elongation. They can use both, actually. And in some ways, de novo synthesis can be used directly from elongation. But coming back to fast 1 is believed to be, at least in toxotachyzoate, inactive during a tachyzoate life stage. And there's like this consensus data coming from Dominic Soldati and Ali Hakimi that it seems could be more active during the bradyzoite life stage. So the current consensus, which is not fully tested, and I've tested at the end of the question, I tested the activity of FAS1 during tachyzoate, and it doesn't seem to be indeed active, but it may be more active during bradyzoite, low division, and FAS2 is the booster kind of thing.
Okay, so Pawan, who is it with Pushkar at the moment, has a question.
Cyril.
Yes.
Hi,
Pawan.
How are things? I am with our friend Pushkar.
I see that.
Yeah, you have to keep the
I am jealous not to be there with you and having some of the wonderful food in the backyard there.
As soon as something Corona comes down, please do visit us. It will be very nice.
I promise I am coming. You don't even have to beg. I am coming. Whatever, you pay me or not, I'm coming.
And I have the 3X with me. That's
Okay, so thank you, Cyril. Thank you once again. Thank you very much.
Thank you all. Please do not hesitate to ask more questions. I'll be happy to answer. And if you want to study with us, get in touch with Asif, Pushkar, and Pawan. Maybe there's a project looming somewhere.
Okay, great. Okay, our next speaker is Dr. Arun Kota. I'm not sure if he's joined. Arun, have you joined?
I'm here, Asif. You're able to hear me?